Hey everyone, it's Andrew. Today we are going to be doing a mini prep. So a mini prep is a protocol that we use to isolate DNA from bacteria, in this case E. coli. And E. coli in this case are acting as our little factories for producing and replicating our plasmid DNA, which we're then going to harvest and use for our downstream purposes. So I just pulled this block, 24-well block, from this nice Inforce shaker that we have here. You don't need a fancy shaker like this. It has a very short distance throwing arm and allows you to shake at really high RPM, which helps the E. coli grow in these blocks, but you don't actually need a shaker that goes all the way up to 750 RPM. Even something at like 250 RPM will still be fine. And actually I found that us growing our E. coli in these 24-well blocks is somewhat unique. I think most people are typically used to growing their E. coli in individual culture tubes, but since we do a lot of cloning here in our lab, I've found that these 24-well blocks help us save a lot of time. Instead of having to manually handle 24 tubes, I can just grab this one block that has enough volume for room in there for 24 cultures. So what I'm doing now is I'm preparing a balance for my 24-well block, so I can actually spin down our E. coli and remove the supernatant, or the media in this case, which is step number 1. Another great thing about these 24-well blocks is they can be washed, autoclaved, and reused, and that helps us cut down on all the waste that we generate in lab on a regular basis. Now, if I'm going to be just growing like a couple cultures at a time rather than the what do I have here, 15 on this block, then something you can do is grow just a few cultures at a time, let's say like in these wells of this block, and then just keep the rest of these wells here kind of sterile and unused. Well, I guess not so sterile, but unused, and then the next time you go around and want to grow some more bacteria, you can just grab this same block and then use up the unused wells, as long as you are good about kind of like keeping it sealed and marking which wells are used versus not. Typically the antibiotics in the media will kill off any unwanted visitors that we might have otherwise floating around in the block or in the wells. Okay, so now I'm gonna spin for 5 minutes at 4,000 RPM in this centrifuge, and we'll come back in 5 minutes to pull out our block and dump out the media. And in the meantime, let's go prep some of our other reagents to do our mini prep. So of course, you always want to make sure that the centrifuge comes up to max speed okay before walking away. And we're looking good, so let's move on. So there are a variety of different mini prep kits out there. Most of them are about the same. In our lab we use a kit that is sold by Thermo Fisher. Actually, I think this is a mini prep kit right here. Yeah, we use the GeneJet mini prep kit from Thermo Fisher. This works well for us. It's a little cheaper than what some might consider to be like the gold standard for mini prep kits, which is the QIAGEN kit. So that's a big plus because of course we burn through a ton of mini preps in our lab. There is only one scenario in which I've found the QIAGEN kits to be superior, and that's when purifying plasmids that contain like a lot of GFP expression. So we use some backbones that express GFP as a dropout for cloning purposes. And we found that for whatever reason, the Thermo Fisher kits tend to purify a lot of that GFP in the eluent, whereas the Kyogen kits don't. And that somehow all of that extra protein messes up our downstream cloning reactions. So I recommend using the more expensive Kyogen kits for things like prepping these vectors that have a lot of GFP production in them. But otherwise, the Thermo Fisher kits work just fine for us. So what I'm doing now is I'm laying out 1.5 mil Eppendorf tubes in this rack in the shape and orientation of the 24-well block. So the way I orient my tubes in this rack is each row corresponds to a row in the 24-well block. This would correspond to the first column of the 24-well block. Then I'll skip 2 columns to allow enough room for my fingers to make it in here and close and open these tubes. So each, what is this, 4th column will correspond to 1 column in the 24-well block, and it just so perfectly happens that there are 24 columns on this very standard tube rack, so it's kind of perfectly in the shape to be mapped onto this 24-well block that we're used to using. So what else? So we need to label our tubes. So rather than giving them the name of the plasmid, which is a huge pain because sometimes, you know, the names of these plasmids can be very long and complex, and writing 15 to 24 labels on tubes, or if you're doing even more than that, can be a huge time suck. So instead, what I'm going to do is just label these tubes 1 through 15 or 24, however many plasmids you have. And just know that when I go ahead and transfer my E. coli from the block into these tubes, that there's kind of like a set mapping that I'll do that in. So I always go from left to right, going down each column before moving on to the next one. So as long as you have your own favorite mapping in place, I know some people in our lab like to go horizontally rather than vertically, so they would label 1, 2, 3, 4, 5, 6 rather than 1, 2, 3, 4, 5, 6. It doesn't really matter as long as you personally know which mapping you like. So the other things I'm going to gather are our buffers. So I'll explain in a second what each of the steps of the mini prep are. Here we go. And the steps are going to be resuspension, lysis, neutralization, binding, wash, and then elution. And this is all, of course, mapped out in great detail in any protocol for mini preps that you might commonly use. And so, perfect timing, the centrifuge is just about done spinning here. So as I was saying before, what we're going to do now is take this block and let's see if you can actually see what this looks like. So you should be able to tell now that you can see the cultures are all pelleted at the bottom. You always want to visually inspect to make sure that your cultures have properly pelleted before throwing away that supername, because yeah, that'd be a big shame if you lost all of your cells in the process of doing this. So now that I know my cells have been pelleted, I can take the media, which yeah, shouldn't contain any E. coli in it now, and dump that down the drain, since this is just BSL-1 stuff, so it's really nothing to be worried about. I'm just going to take this, invert it in one swift motion, give it another toss for good measure. And then here's a trick, is to actually take the block now and dab it on a paper towel to absorb any of that excess moisture, which otherwise would have drained back into the bottom of our wells. So as you can see, there actually is quite a lot of leftover media that hangs around like the lip of each of the wells. So now I can take this, throw this in the trash. And another key thing to remember is you need to save this little piece of film on top of which we've written exactly what plasmid is in each well, because, well, if I toss this, then I'll have no way of knowing what plasmid actually lives in which well, and that would be bad. Again, big loss of work for us. All right, so let's see how we're doing here. Okay, so as I said before, the steps for this protocol are resuspension, in which we're going to take these nice little pellets of E. coli that we have and resuspend them in this buffer, which contains RNase A to degrade any RNA that might be floating around. Since this is a DNA isolation, we don't want to isolate any RNA on accident. Once I resuspend each of these cultures in the block, I'm going to transfer them now into the Eppendorf tubes in this rack, and then I'm going to lyse the cells. So I'm going to pop open all of those E. coli so that they spill their guts, and inside those cells are going to be our plasmids, which we want to eventually purify, but there's also a ton of other crap in there as well. You know, all the stuff that helps the cell live, like all of the proteins, of course the bacterial DNA as well. And something that we want to be extra careful of is, as we do the lysis, well, I'll explain more when we get to this step, but we don't want to lyse the cells so hard that we actually shear the bacterial genome, because of course if we shear the bacterial genome, then we're going to end up with a ton of bacterial DNA in our Minipreps. Luckily, the Miniprep developers were smart enough to develop these columns that would only elute DNA of a certain size, and so if you don't shear the bacterial genomes, then they won't bind the column. But if you do, then of course you're breaking the genome down into much smaller pieces of DNA, which then are able to bind the column and then eventually get eluted later. So here, because I have 15 samples, I'm actually going to use this handy dandy repeater pipette. And what this repeater pipette lets me do is just save a whole bunch of steps of kind of going back and forth, transferring tips and whatnot to fill up my block with resuspension buffer. So I've set this repeater pipette to 250 microliters, and I'm using this combi tip, which I actually just store here and reuse. We try and replace these every few weeks or so. They get quite gross after a bit. But yeah, they can definitely be reused for a little while. So what I'm going to do is aspirate my resuspension buffer, which, as I said before, contains RNase A, which you have to add yourself and then store at 4 degrees. And now that I've aspirated my resuspension buffer, I'm going to simply transfer this into the block, being careful to, 1, not touch the combi tip to any surface on the 24-well block. And you might also notice that I'm shooting the resuspension buffer actually onto the sides or the corner of the block to prevent any sort of splashing that might occur if I were to just directly shoot the resuspension buffer onto the bottom of each well. And so since I always do a very careful job of making sure this tip doesn't touch any other surface, I can just then go ahead and shoot any excess that I might have back into this bottle. Now, if you're feeling paranoid or not sure if you touched any surface, then of course you wouldn't want to do that, and also you would want to throw away your combi tip. So this is going to go back into the 4-degree fridge where that lives. And now what I'm going to do is recap this block with the breathable film, make sure all of the wells are nice and sealed, and I'm going to vortex this to resuspend all of my E. coli into the resuspension buffer. Usually this just takes 10 to 15 seconds or so. And voila, hopefully you should be able to see that all of those pellets, which we previously saw, are now resuspended into solution. So now I'm going to take my P1000 and just simply transfer each of the wells in my 24 well block to the corresponding well or corresponding tube in my tube rack. Yeah, so each of these has been nicely resuspended, and you'll notice, well, maybe it's a little hard to see, but even after pipetting 250 mils, there is a little bit extra left over. So what I will do is actually press down to the second stop on my pipette to make sure I get all of that resuspended E. coli that I want from each of these wells. So let's see, what else? So what I didn't show you last night is me actually picking colonies into this block. So I can just very quickly explain how I do that. It's very simple. You fill first each well of this block with 3 mils of LB plus the appropriate antibiotic for performing the selection against the plasmid that you want to grow inside that well. And then you can just take a pipette tip. Usually I'll use a P200 pipette tip, one of the pipette tips from this green box, and scrape off a colony of your E. coli. Scrape it around the well into the media, and yeah, that's it. It's very simple. Just pull out those pipette tips when you're done, seal with one of these breathable seals, and pop it in the shaker overnight. And by overnight, I kind of mean a full range of times actually. So depending on the strain of E. coli that you're growing, you will need a different amount of time to really reach saturation of your culture. So we used to work with this strain of E. coli called Mach 1 that would actually saturate in about 8 or so hours if you were growing in carb or amp antibiotic, which was really great for some protocols. But for these slower growing E. coli that I'm using now, these stable 3s, they definitely take at least 16 hours to reach saturation. So I picked colonies yesterday around 5 p.m. Right now it's almost noon, so that's about 19 hours. I guess the earliest that I would have recommended starting this mini prep would have been 16 hours, which just so happens to be around 9 a.m. Now, how long would I be willing to grow this before I'd be worried about the E. coli starting to degrade some of the plasmid DNA that's actually in here? Maybe about 24 hours would be the time limit. I think if you anticipate not being able to do your mini prep within 24 hours, what you can do is just pull this block out and either directly put it in the fridge if you're in a big rush, or better yet, what you can do is spin the block down, dump out the media, and then just freeze that block at -20 until you're ready to do your mini prep. Okay, so now we have all of our E. coli transferred into this block. So I'm going to grab my combi tip, my lysis combi tip, and do the same thing as I did before. Aspirate a bunch of this lysis buffer, transfer into the tubes. Again, I'm not sure if you're able to see this or not, but I'm trying to be very careful to not touch the tip to any of these tubes, and also shooting the lysis solution against the walls of the tubes to make sure I don't get any unwanted splashing. Okay, so as I was mentioning before, we kind of want this fine line of lysing the bacteria hard enough so that they release all their contents, but not lysing them so hard that all of the bacterial genomes actually fall apart and we get a bunch of bacterial DNA in our eventual eluent. And so the way I like to lyse is by taking a second one of these tube racks and just placing it on top of my tube rack once I've capped all of the tubes, and just make sure they're all capped very tightly when you do that. And then just giving it a bunch of inversions, somewhere between 10 and 20. And then maybe a couple shakes for good measure. So now that the lysis is complete, I know some kits will come with some sort of like blue solution that once all the bacteria are like fully lysed, the blue goes away. So that's kind of cool if you have that, but if not, then you just kind of have to try it a couple times and see how your mini prep yields are to know whether or not you are doing a good job of lysing your cells without breaking down those bacterial genomes. So manufacturers typically don't recommend that you let your cells sit in lysis buffer for too long. So now I'm going to very quickly add the neutralization buffer, which stops that lysis process from happening. Now I'm not really talking too much about like volumes and times here because all that information is written down in the protocol, and so this is definitely not a substitute for the written protocol. It's more to give some color and kind of like tips around the written protocol. So I definitely recommend that you still read the written protocol and have that at your side while performing any protocol such as this one. But feel free to use this video and this video series as kind of like a study guide, something that will help you maybe understand why certain parts of the protocol work, or certain things that you might want to watch out for that are not necessarily written down in great detail. So what I'm doing now is, again, just repeating the last step that I did with the neutralization buffer now. I'm just trying to mix the neutralization buffer very thoroughly in each of these tubes to stop the reaction. So now you'll see that the tubes have gone from clear looking to this milky white looking thing. And so all of this kind of debris that you see in this tube, well, that's our E. coli. Those are the bits of the cells, the proteins that were previously part of the E. coli and have now just been broken apart into billions and billions of little pieces. So now what we're going to do to actually separate our DNA, which is not part of this debris but is actually floating around in the liquid part of this tube, is we're going to centrifuge our tubes for 5 minutes at max speed to separate all of that debris from the plasmid DNA, which we want And so as I'm adding these tubes to the centrifuge, I'm also just looking to make sure that they are well mixed before throwing them in. And if not, giving them a little shake. Okay, let's make sure I can balance the centrifuge properly. And here, as before, I'm just going to have to remember that I need to replace my tubes back onto my rack in numerical order later to retain my mapping. Okay, so I'm going to bring this puppy up to max speed, 15,000 RPM, and we're going to spin for 5 minutes. Great. So during this time, I can prepare for the next step, which is going to be binding onto these Miniprep columns. So these Miniprep columns have a few different parts. This pops off right here. So this tube is just meant to catch all of our wash, buffer, runoff from the supernatant that we're about to pull off from these Eppendorf tubes that are spinning now. So this is just basically a waste receptacle. This is the actual Miniprep column itself. Has a little lid on it, which I personally don't think is that necessary. And here you see this kind of like this piece of paper here. This filter is actually the thing that is going to absorb our DNA. So be very careful when you're pipetting into this Miniprep column to never pierce or touch this filter, because if you do, then you're going to mess up this column. So just as before, what I'm going to do is lay out my tubes in this tube rack in the same orientation as before. If I'm lucky, then it's annoying. I have exactly 14 columns and I need 15. Luckily, we have a bunch of kits floating around here. There we go. And as before, I'm going to label these tubes now. 1 through 15. Something that I think is really good to get in the habit of as you're doing molecular biology, but really anything in science, is to always think ahead to the next step. What's the next step of my protocol? And always trying to prepare yourself for that step. So whether that be gathering reagents or supplies ahead of time to make sure you have everything that you need before starting, or during one of these long either incubation steps or spinning steps, utilizing that time to your advantage to prepare for the next step in the protocol. I remember when I first started out doing lab work, I would always think, oh, okay, like I have 5 minutes now, I can step away from the bench for a little bit. Someone took out these little stoppers, and so I now have to put them all back onto the vacuum manifold. But actually this time that I'm waiting for my tubes to spin down is a perfect opportunity to prepare for the next step and save myself time in the future. So what I just set up is this thing called a vacuum manifold. It hooks up to our house vacuum line over here, which you can see. There we go. And the idea is that my Miniprep columns should fit nicely into each of these little valves. Such that when my centrifuge is done spinning, I can transfer the supernatant into these tubes And rather than using these as our waste receptacles, which the protocol calls for you to do a series of spins and emptying these tubes, which takes a lot of time, running your tubes on a vacuum manifold is much faster because you don't have to actually go through the process of spinning your tubes and taking them out of the centrifuge, emptying the waste containers. So if you do a lot of mini preps, then I think a vacuum manifold is a great thing to invest in. You just have to be sure to clean them every now and again, and otherwise they're cheap, low maintenance. I don't see any reason why you wouldn't want to use them. So you might be able to hear that the centrifuge has just finished. So in my last remaining seconds, I'm going to try and open up all of these valves that have tubes on them and keep the others closed. So we were just a little bit slow there, but close enough. Okay, so now I'm going to rescue my tubes from the centrifuge. And you can see that all of that debris that was previously floating around quite homogeneously in the tube has now formed a nice pellet on the side here. So this white pellet is all of the stuff that we don't want. All of the cell debris, protein, hopefully the bacterial genome is in there too, and all of this nice supernatant contains our plasmid DNA in there now, and we want to bind that onto our mini prep columns. If for whatever reason you don't see a nice pellet like this, then you can try spinning your centrifuge again. I've found that these 1.5 mil Eppendorf tubes make a nicer pellet than 2 mil Eppendorf tubes, which would be the other more commonly used tube for this protocol. So that's why I opt to use this kind of tube, but you don't have to. So I'm almost out of P1000 tips here. Looks like there's another box right here, so I'm ready to go. Okay, so previously we used 250 microliters of resuspension, 250 microliters of lysis, and 350 microliters of neutralization buffer, so that adds up to 850 microliters. So I've set my P1000 pipette to 850 microliters, and now I'm just going to transfer all the supernatant into these columns to bind the plasmid DNA to the column like we were just talking about. So here, as you're pipetting, be careful to not disturb that pellet, and just visually examine your pipette to make sure that the supernatant is clear. And if it's not clear, then just spin your columns again. So again, very easy. All I'm doing is transferring the supernatant onto these columns. Be very careful here to not toss your supernatant. The supernatant has all the good stuff. In the next few steps, after we add wash buffer to these tubes, and especially if you're not doing this on a vacuum manifold, we're going to want to discard that wash buffer and keep what's bound to the column. So you just have to remember at which steps in the protocol, what is the thing that I want to keep. In this case, we want to keep the supernatant on the column as it binds. Looks like these tips are actually the wrong fit for my pipette, which is a little annoying. Let's see, are these P1000 tips? Okay, I'm going to be right back while I search for some P1000 tips. Okay, we're back. I have located some P1000 tips. and we can continue. Okay, on to the next step. So now that I have transferred all of the supernaine into the tubes, I'm going to run the vacuum, and you'll see that the supernaine is being slowly drawn through each of these tubes. There you go. Hopefully you can see that the water level is just slowly dropping in each of these. And once that is fully drawn out, I'm going to take my wash buffer and this big combi tip here and use my repeater pipette to now transfer 1 ml of wash buffer into each of these tubes. Now this wash buffer is mostly ethanol, so the DNA will not elute in this wash buffer. Instead, we're just getting rid of any other impurities that might be stuck to the column.
So again, this wash buffer will help us get rid of any other junk that might be floating around in the tube or bound to our column, but will not elute the DNA, so we are good to throw away this wash buffer. And
after all of this wash buffer has been drawn out of our manifold, what we can do is shut off the vacuum and then transfer these tubes into the little waste receptacles that they came in. And I'm going to give it a quick spin on the centrifuge now to remove any of the remaining wash buffer that the vacuum really isn't strong enough to suck through. And you might also just be able to see that there is kind of some stuff that just gets stuck to the walls of this tube that are really hard for the vacuum to do a good job of fully cleaning. So all we need is a quick spin in the centrifuge, like 1 minute. And this is a key step because otherwise, when you elute your DNA in the next step, you are going to have a lot of ethanol in your mini prep. And we don't want that, especially if you are doing things like using this DNA for transfecting mammalian cells, because that ethanol is going to really mess up your cells and probably just mess up whatever assay you're going to run with this DNA downstream. So as I said, quick spin, and during this time we can take our tubes that originally contained the E. coli cultures that we then lysed and spun down, toss those because we don't need them anymore. And I'm going to grab a fresh set of 1.5 ml Epindor tubes. Now I'm going to use these tubes that were actually autoclaved because I just want to be sure that these tubes are very clean for storing our DNA, which is going to go and be transfected into human cells. So I'm going to lay them out in this rack as before, and I'm not going to label them yet, and you'll see in a second why. Alright, our quick spin is done, and I'm going to show you in the waste column actually how much ethanol was left over in those tubes, even though at first glance on the vacuum manifold it might actually have looked like they were empty. So there's number 1. Here we go. Yeah, you see that? So there's actually quite a lot of ethanol that you see at the bottom of the tube. So just be careful when you're removing these not to like jostle the tube around too much, or else that ethanol is going to end up back on the column, and we want our column nice and dry for this last step. Okay. Again, I'm just double checking to make sure everything is in numerical order here. Perfect. Now I'm going to simply transfer these dried columns onto my fresh Eppendorf tubes. And then I'm going to add my elution buffer, which is mostly water, plus some salts, like I think EDTA or something is in elution buffer. Can't remember off the top of my head. You can actually just use water to eluten if you want. Alright, so I need to pop open these columns. And I like to elute in 60 microliters. Usually protocols will have 50 microliters written down. I haven't found that with an increase of 10 microliters that my concentration actually goes down. There's this kind of trade-off between eluting all of the DNA off of the column, but also making sure your DNA concentration is high enough. So protocols will recommend anywhere between 50 and 100 microliters of DNA. I like my DNA to be concentrated for doing transfections, so 60 is kind of a good intermediate for me. And you can do this step with the repeater pipette. Here I don't. I do it manually, but with a single tip. Again, super careful not to touch anything with this tip except the elution buffer. Shooting the elution buffer onto the sides of the column to prevent splashing. And the reason why I don't like using a combi tip for this step is because, well, this is really the most sensitive to any sort of contamination. I don't want to elute any random crap into my tube, I just want to elute the thing that I came here for. So I use a fresh tip every time. And if you have one of these fancy electronic pipettes, then you can also use that. So here at this step, again, some protocols will recommend a few different hacks to maximize your DNA yield. Incubating the columns with the elution buffer on them for anywhere from like 1 to 10 minutes. Another trick is using warm elution buffer. So something some people like to do to really maximize their yields is take an aliquot of this elution buffer, maybe like a mil or something, and then warm that up to somewhere between like 40 and 50 degrees Celsius. And that just helps speed up the reaction of the transfer of DNA from the column into our elution buffer, or EB. I've found that both of those steps aren't really necessary. In an ideal scenario, your column should just be like super saturated with plasmid DNA anyways, so it doesn't really matter that much. So I'm gonna go ahead now. It's probably already been about a minute. Transfer these columns into the centrifuge, just as before. Here, the only difference is these lids get in the way and make it annoying to load all of the tubes in. And so I've found it's actually quite difficult. Let's say if you want to load up like a full set of 24 tubes into the centrifuge, it's pretty tough. I think max you can get about like 20 into a centrifuge of this side, and that even might be pushing it a little. Let's see, am I off here? Yeah, I'm off by 1. So if I'm doing a full set of 24 mini preps, I'll actually just usually split that in between 2 centrifuges. We're lucky enough to have a second centrifuge right behind us that I can use. So yeah, you can see already these like lids are kind of bumping into each other and giving me a headache. The other thing that I want to do is drop down the speed of the centrifuge. If I were to spin it at 1500 RPM, I would actually just snap the lids off of those Eppendorf tubes right off, which would be a pain for several reasons. So instead I'm going to spin at 8,000 RPM, which is fast enough to elute everything in a minute, but also won't snap the caps off my tubes. And that's it. There you go. That is the full extent on how to do a mini prep. After these tubes come out, I'm going to remove the mini prep columns. I'm going to save them just in case for whatever reason. I don't know, somehow I might have forgotten to pipette elution buffer into a column or something. I'm just going to keep them over there while I go ahead and verify my plasmids via a restriction enzyme digest and nanodrop to check their concentration, after which point I will feel comfortable throwing away those columns. So yeah, it's a super simple protocol. It takes about half an hour to 45 minutes, depending on how fast you are, to do about 24 mini preps, kind of given the workflow that I've outlined for you today, using a combination of the 24-well block and a vacuum manifold. So when you are preparing to do this protocol yourself, just know that that's about how much time you should budget to get it done. Okay. And here you can see now that we've eluted a small amount of liquid into the bottom of this tube. That is our plasmid. Hopefully a nice and pure prep of the plasmid that I want at the bottom of that tube. And same as before, what I'm going to do is, once I get all my tubes out of my centrifuge, oops, and you can actually see that it's a good thing I've labeled these because I went a little out of order here. 10, 11. Now, take the columns out of my tubes. And then again, give the tops of these a very quick label, 1 through 15. Again, making sure that I've kept this block here with the writing on top so I know the mapping of what number is what. Very important to not throw that away just yet. Alright, and there you have it. That is how you do a mini prep. Alright, thanks for watching.